Okay, this chapter deals with adaptive immunity, which is our ability to clear an infection as a result of the presentation of an antigen, which is a foreign molecule on the surface of the pathogen, unlike our own. Okay, it's inducible, it's specific, it's long lived, and it features memory. The elements are antigens, which can be any surface molecule, um, anything ranging from a protein to a glycoprotein to a glycolipid to a carbohydrate. It really doesn't matter. Antigens just need to meet a certain size and diversity standard in order for adaptive immunity to recognize it and clear it. And then it involves lymphocytes, T and B lymphocytes. Okay, T lymphocytes come in a variety of classes: helper Ts, memory Ts, suppressor Ts, and cytotoxic Ts. And then B cells, which become plasma cells that produce antibody. The components are humoral immunity, which utilizes antibody to bind to and clear antigens, and cellular immunity, which uses cytotoxic T cells to destroy infected cells and tissues that present foreign antigen on their surface. Both produce memory and they interact with each other. Okay. In active immunity, um, we have either natural active or artificial active immunity. In natural active immunity, you acquire the infection and then clear it. Okay. And as a result, you generate memory lymphocytes that stay with you your whole life. And in artificial active immunity, which is immunization, you get an injection of antigen, which you then generate a response to that produces memory. And then, in the event that you encounter a pathogen that bears those antigens, you're able to clear it with a secondary immune response. In passive immunity, you acquire antibodies from another source. So, this is temporary protection while the antibodies are in your system. Natural passive immunity is exemplified, such as when the fetus is getting antibody through cord blood from the mother or through colostrum or breast milk from the mother. And artificial passive immunity is injection of antibody through a needle. Okay, antigens bind antibodies and also receptors on T and B cells. They may or may not be immunogenic. They have to meet a certain size and diversity standards. Okay, immunogens induce production of antibodies, um, as well as the differentiation and propagation of certain T and B cells. All immunogens are antigens, but not all antigens are immunogens. And a hapten is simply a foreign molecule too small to be immunogenic on its own, but in the context of a larger molecule, it is immunogenic. You can see here that there are a variety of antibody classes defined by their constant regions. Okay, now what we have to understand about an antibody is that there is an antigen binding region, which are the tips of the Y-shaped portion of the molecule. Let me pull those up here if I can. Oh, really? You're gonna give me problems, huh? Well, all right. The tips of the Y-shaped molecule are where the antigen binding takes place. Let's circle those. Okay. So for instance, the antigen binding site here would be same as the antigen binding site here and so on. But it's the constant region, which is essentially, let's circle that in a different color. The constant region, which defines where the antibody does its job. Okay, this would be a constant region. Okay, this will be a constant region. Okay, and so on. All right, so these proteins have both constant and variable regions. All right, I'm circling all these constant regions here. The constant regions determine where the antibodies function, right? And the variable regions, the tips of the Y-shaped molecule, determine what it binds to. Okay, let's just go ahead and finish out what we're doing here. Okay, so here is a antigen binding region, here is one. The antigen binding regions on a given antibody are identical, so you can engage two antigens at the same time per monomer, right? And then I won't circle all these. That's a pentamer. Each of these are antigen binding regions here, okay? And the constant region is the core of this pentagon, okay? All right, circle that. This is all constant region. Okay. Now, what does what where? Well, IgG and IgM are found in plasma, okay, and they're capable of binding complement and clearing antigen, okay. IgA is found in secretions, all right, and such as saliva and milk and colostrum and sweat, uh, you know, and oil, and can bind antigen but cannot bind complement. IgE binds antigen and then also binds receptors on mast cells and basophils to trigger histamine release. And IgD is actually B cell receptor and is not secreted. Okay. IgG is the most abundant class and accounts for most of the protective activity against infection. It's transported across the placenta. The classes are IgG 1 through 4. All right. IgA has two classes found mostly in blood. IgA2 are found predominantly in bodily secretions, okay, which is its most important role. Secretory IgA is anchored by a J chain, as we saw in the previous slide, and the secretory piece may function to protect IgAs against enzyme breakdown, okay, but it does not bind complement, all right. IgM is the largest of the immunoglobulins. It's a pentamer stabilized by J chain. It's the first antibody produced during a primary response to an antigen and made early in neonatal life and can bind complement. Okay, so generally, when we have an antibody response, IgM is first, IgG follows. Okay, IgD is a B cell receptor. Okay, and is not generally found free floating in the blood or in the fluids. Okay, the antigen binding fragment has the recognition sites for the antigens. The constant region is responsible for determining where the antibody functions. There are two light and two heavy chains that make up the structure. The constant regions are stable amino acid sequence. The variable regions have large numbers of amino acid differences. You might wonder how many. Between a billion and a trillion possible different sequences, depending on the individual. Okay. The complementarity determining regions and the framework regions are all shown here. Okay, so you see again that the antigen binding sites are at the tips of the Y-shaped molecule. They engage the antigen, the target, and that tags it for destruction in a variety of ways. Okay, um, complement usually binds antibody bound to antigen. Okay, and that helps to clear it by promoting a wide variety of responses, such as generating membrane attack complexes and opsonization and attracting neutrophils, promoting inflammation. Okay, all of these things we'll see, in addition to things like neutralization of toxins and coating of viruses to prevent penetration into host targets. Okay. Antigenic determinants are the area of the antigen recognized by the antibody, and the antigen binding site is the matching portion on the antibody. The antigen fits like a key into a lock. The specificity is very, very strict. Okay. Antibody functions in neutralization, agglutination, and precipitation. Okay. Those are direct effects, meaning in neutralization, for instance, we are um, coating the surface of the target so that it can't, for instance, be a toxin. Okay. In agglutination, we're clumping cells together so that they can't effectively engage in infection. And in precipitation, we are essentially clumping together things other than cells so that they're ineffective. Okay. Indirect processes promoting inflammation, promoting phagocytosis through opsonization and attraction of neutrophils, and binding complement. Okay, which helps to clear the target that the antibody is bound to. Okay. IgE is the least concentrated of immunoglobulin classes in circulation. It's basically an initiator of inflammation. It defends against parasites. It provides protection from large parasites and initiates an inflammatory reaction to attract eosinophils. when produced against an innocuous environmental antigen, it causes allergies. Okay, the Fc portion of the IgE binds mast cells and basophils and results in histamine release. Okay, so you can see here how the IgE is doing its work, right? He can essentially by binding the target, right? You can see down here, um, he gets produced. Okay, this is the parasite antigen up here. All right, he gets produced by the B cell. He binds the target, which then binds the mast cell, which then releases cytokines and histamine that attracts the eosinophil. Okay, and then the eosinophil is able to move in and attack the parasite. Okay, remember that the job of eosinophils is really twofold: to attack parasites and to eat antigen antibody complexes, and also to turn off inflammation. Okay, because it has antihistamines in its granules. Lymphoid tissues that protect the external surfaces of the body contain secretory IgA. Okay, and there are small amounts of IgG and IgM, but generally you don't want to activate complement in secretory regions. Okay, but the secretory IgA, again, is important in clearing foreign antigen. Okay, so you can see where you end up finding malt. Okay, and where we're able to bind and clear material. So again, IgA in mucus, in saliva, in sweat, in tears, in milk, in colostrum, um, in oil. Okay, all of that. All right, and you can see again the sites of production. Okay, clonal diversity is one of these things that it's difficult to kind of wrap our mind around because we have between a billion and a trillion different specificities. If we were to use a single gene in the nucleus for every single one of the possible antigen binding specificities for antibodies and T cell receptors, we'd more than use up all the DNA in the nucleus. So, how is it that we're able to generate the clonal diversity? And the answer is through germ level recombination of DNA segments um, that involve hundreds of different pieces of DNA that can be combined in any order and can be combined in the face of either removing or adding nucleotides as well. Okay. Um, all necessary receptor specificities are produced in diversity, and this takes place initially in red bone marrow, and then for B cells. Okay, if they bind their appropriate antigen, they're maintained for further development. If it takes in the if it's we're talking about T cells, they're generated in the red marrow. They then move to the thymus and are selected for immunocompetence, and then deployed to the remaining primary lymphatic organs in the body. Okay, so the production here of naive but immunocompetent T and B cells um, takes place. Uh, this is something that's going on beginning in the fetus and through the early stages of neonatality. All right. And then it finishes as we enter childhood and adolescence. Okay. And by that time, the thymus is no longer needed and essentially becomes scar tissue. Okay. B cell development involves production, proliferation, differentiation in the bone marrow. That's why they're called B cells, because they come from the bone marrow. They travel to lymphoid tissue, reside there as immunocompetent cells. Each cell responds to only one specific antigen. T cell development initially in the red marrow, then we move to the thymus. We're selected for immunocompetence. Okay. The thymus is the central lymphoid organ of T cell development. They leave the thymus, travel to and reside in secondary lymphoid tissues and as mature immunocompetent cells. And then they await the arrival of their appropriate antigen. Okay. So the thymus is sort of like T cell school. All right. So one of the things we have to take note of when we're thinking about the amount of diversity that we have, right? We can recognize up to 1 trillion different antigens with our adaptive immunity is the fact that if we were to dedicate all the DNA in the cell to those trillion different specificities, we would more than use up all the genetic material. So the riddle becomes, how do we get to that enormous number of 1 trillion different specificities, meaning 1 trillion different binding sites on those antibody antigen binding regions, or those T cell receptors. And the way that I like to refer to um, this process is to think about the game of poker, right? If you've ever played poker, you have a deck of 52 cards. If I shuffle that deck and deal you 5 cards, how many possible different hands could you get? And the answer is 52 times 51 times 50 times 49 times 48, which is a huge number. Okay. If we take this analogy and we apply it to how we generate the diversity that we see in antibodies and T cell receptors, we do the same thing with pieces of DNA at the germline level. By recombining them and by removing and adding nucleotides during that recombination process, we're able to generate huge numbers of open reading frames that can bind enormous numbers of different potential antigens. So let's look at how this works.
Human cells do not have enough DNA to have separate genes for each antibody molecule. Instead, different segments of DNA can be mixed and matched to form different antibodies. Light chains are made from V and J segments in the variable region and a constant region segment, whereas heavy chains are made from V, D, and J segments in the variable region and one of 5 different constant region segments. As the lymphocyte divides during maturation, its genes are rearranged. DNA sequences are assembled by the random selection of V and J gene segments for the light chains and V, D, and J segments for the heavy chains. The constant region of the molecule is encoded by a single C or constant gene segment for light chains and 5 different constant regions for heavy chains. The rearranged DNA in the mature B cell is transcribed into messenger RNA and translated into the light chains of the antibody. DNA sequences for the variable regions of each heavy chain are assembled by the random selection of a V gene segment, a D gene segment, and a J gene segment. The genes for the constant regions of heavy H chains have Greek letters designating IgG, gamma, IgM, mu, IgA, alpha, IgE, epsilon, and IgD, delta. The DNA in the mature B cell is transcribed and translated into the heavy chain protein. The light chains are then attached to the heavy chains to form the functional antibody. Because of the large number of ways in which the genes can be rearranged, many different antibodies can be produced.
So the statement that all cells in the body have the same DNA sequence front to back, with the exception of your gametes, there's another exception, and that's your lymphocytes, right? The antibody genes and the T cell receptor genes are going to be different from clone to clone to clone because of this recombination process. And that's how we're able to get to between 1 billion and 1 trillion different specificities. That's why we can state that your adaptive immunity in everybody is functionally identical, okay? Functionally identical, but structurally different, okay? Meaning it does the same job, but it's made of different pieces, okay? Your primary immune response it follows your initial exposure to foreign antigen. There's a latent or lag phase where B cell differentiation is occurring. After about a week, IgM antibody for a specific antigen is detected in the plasma, and then an IgG response of equal or slightly less follows the IgM response. During this process, you make memory cells that stay with you your entire life, and with a second exposure, all right, it's a more rapid response. It is orders of magnitude more rapid and orders of magnitude more efficacious, meaning there's more antibody produced, okay? The rapidity is the result of the memory cells that require less differentiation. IgM is transiently produced, but IgG is now the predominant antibody in your plasma to clear foreign antigen. Antigen processing is initiated when T and B cells interact with antigen. They have to be presented on an antigen-presenting cell, resulting in differentiation of B cells into active antibody-producing cells and differentiation of T cells into effector cells, such as cytotoxic Ts. Okay, now most of this involves interaction with what's called a helper T cell. Okay, helper T cells are going to be activated when they encounter on an antigen presenting cell the antigen of their specificity. Okay, and then they're going to interact directly with naive B cells that recognize that same antigen, and they form a complex that involves T cell receptor from the helper T, and then MHC2 from the B cell, and then CD4, all right, or yeah, I believe that's right. Yeah, CD4 from the B cell as well. And when they interact, these membrane proteins then stimulate that B cell clone to grow and divide and make a little army that become plasma cells and memory cells. And that's the humoral part of the immune complex. Okay. Now, T cells are a little bit different. Helper T cells stimulate cytotoxic T cells through diffusible elements called cytokines that activate them after they've bound their presented antigen on um, a helper on an antigen presenting cell like a macrophage, and the result then is they become fully activated and they produce a little army that goes out and kills cells that have foreign antigen on them, uh, essentially processed and presented with MHC1. Okay. MHC is basically like an hors d'oeuvre tray if the antigen is the hors d'oeuvre. Okay. It's on the surface of all human cells except red cells. It's also referred to as HLA antigen. Class 1 presents endogenous antigens, while class 2 is exogenous antigens. Exogenous antigens come from outside the cell and get presented, while endogenous antigens are produced inside the cell and presented. Okay, so you can see where these locuses are located, okay, and where each of these proteins are found, okay. And the way this operates, okay, antigen processing and presentation, um, with endogenous processing, okay, we generate the polypeptide inside the cell, all right. Antigenic peptides from the cytoplasm combine with it and they make a complex, all right. And then that gets presented with a class 1 MHC on the cell surface, okay. With exogenous, what happens is that we phagocytize the bacteria, we bust it up with a phagolysosome, and we take fragments of that and present it with a class 2. Okay. Class 2 with presented MHC on its surface, all right, from an antigen presenting cell is a way to alert the lymphocytes that recognize that to persist, divide, and grow and make a clone army. Okay. Class 1 MHC would normally present self antigen, okay, but in an infected cell, all right, if class 1 MHC presents foreign antigen, that's a signal for that cell to be destroyed, all right. Intercellular collaborations resulting in production of effector and memory cells are critical in the immune response. It requires three complementary intracellular signaling events: antigen-specific recognition through T cell receptor complexes, activation of intercellular adhesion molecules, and response to cytokines. Help helper T lymphocytes are going to stimulate both humoral and cell-mediated immunity. They facilitate and magnify the interaction between antigen-presenting cells and immunocompetent lymphocytes. Th1 cells provide help in developing cell-mediated immunity, and Th2 in developing humoral immunity. Humoral meaning antibody-mediated, cell-mediated meaning cytotoxic T cells. Okay, the differences are related to cytokine production. Note that the Th2 cells physically interact with the B cells, while the Th1s do it through diffusible cytokines. Superantigens bind the variable portion of a T cell receptor in the MHC class II molecules outside their antigen presentation sites. They activate a large population of T lymphocytes regardless of their specificity and induce an excessive production of cytokines, causing fever, a drop in blood pressure, and potentially anaphylactic shock. So this is one of the strategies that are used by certain pathogens to produce disease, okay, which is basically to distract the immune system to the point of damaging you, while the pathogen is able to go off and basically make its living, okay. B cell activation happens when an immunocompetent B cell encounters an antigen for the first time. The B cells with specific B cell receptors are stimulated to differentiate and proliferate. A differentiated B cell becomes a plasma cell, which is a factory for antibody production, producing a class or subclass of antibodies, and is capable of class switching. So if you look here, all right, you can see how this works, all right. If we start here at the beginning, the B cell encounters its antigen. Now, this can be, you know, out in the open, or this can be from a helper T, okay. And then what will happen is that I'm sorry, it could be out in the open, or it can be on an antigen presenting cell, an APC. So then the B cell receptor internalizes. We present the foreign antigen on a class 2 MHC, which then interacts with T cell receptor and CD4 on the helper T cell. See how they physically interact, and CD4 is a big part of this. Interleukins then stimulate the differentiation of this bound B cell into a plasma cell, and then we produce large amounts of antibody. So without the T helper cell, the humoral immune response is muted and relatively ineffective. So that's a very important point to understand, right? T cell activation, we bind the antigen specific to the T cell receptors, allowing direct killing of the foreign or abnormal cell and helping with activation of other cells. Tregs, regulatory Ts, turn off the immune response and avoid autoimmunity. And of course, memory Ts stay with you your entire life, so that in the event that you encounter that antigen again, you're able to clear it more effectively. Cytotoxic Ts are there to destroy infected cells and tissues that present foreign antigen on MHC1. Okay, they destroy cancer cells or cells that are virally infected using perforin, granzymes, or direct receptor interactions. Basically, they punch holes in the membrane, spray it with chemicals that induce apoptosis, and the cell dies. Okay, natural killers also destroy cancer cells. Okay, lymphokine secreting T cells amplify inflammation, and T regulatory lymphocytes provide peripheral tolerance and suppress the immune response. So you can see here again the different mechanisms, right? Cytotoxic T, we engage foreign antigen on MHC1. We induce apoptosis by punching holes in the membrane and hitting it with apoptosis-inducing chemicals, okay? Here we have, again, antigen binding antibody and binding the binding to the T cell, the cytotoxic, right? And inducing apoptosis with abnormal, um, again, abnormal antigen on its surface, okay? And then here you have killing by natural killer cell, right? Abnormal surface change results in binding of natural killer and inducing apoptosis, again, by punching holes in it, right? So here the abnormal antigen is engaged by antibody bound to surface receptor on these cells. Okay, whoops, back up. Okay, and here it's cytotoxic T, and here it's natural killer. Right. Fetuses have sufficient IgM but lack IgG or IgA response because their immune system isn't yet fully mature. Maternal antibodies provide protection in the fetal circulation during the first months of life, and that comes from, again, cord blood, colostrum, and breast milk. Immunologically immature when born, deficiencies in antibody production, phagocytic activity, and complement activity eventually mature. Okay, but this is one of the reasons why babies that are nursed tend to do better with infection than babies that are brought up with formula. Right. As we age, our thymus is going to decrease in size and activity. Okay, thymic hormone production stops, as does the organ's ability to mediate T cell differentiation, and decreased antibody response to antigen results, again, because there basically are fewer lymphocytes to do the job, including helper Ts. And so this is one of the reasons that as we age, we get sicker quicker, and it takes longer to clear infection. Okay. All right. I thank everybody. I'll join you in the next podcast.